This method can help answer key questions about the nature of protein interactomes. The main advantage of this technique is that all the bioinformatics processing is operated by an easy-to-use interface.
This software employs a user-friendly graphic user interface that molecular biologists, cell biologists, and biochemists that don't have a lot of expertise in bioinformatics can use to easily complete their analysis.
Generally, individuals new to processing sequence data struggle because the computer programs are confusing and cumbersome. We thought making the informatics easy would make the whole technique much more accessible and useful.
The first step in this procedure is to download and install the Mapster software program as described in the text protocol. Next, enter the required files and parameters through the Main tab. Select the appropriate pairwise button to enter the files. For deep-in analysis, turn the pairwise option to off to run in single-read format. Load files into Mapster by drag and drop into the appropriate window. From the index genomes listed in the genome box, select a reference DNA genome source that corresponds to the source of the Y2H Prey library inserts. Since HIS-AT2 supports multi-threading, under the Threads box, indicate the number of computer processes to be devoted to the mapping program. Specify an output file name. A short yet descriptive name without space or special characters is recommended since this file name will be used throughout the deep-in process. Using the Open Output Directory button, specify a folder to output the mapped files. Once the